So basically for the non-mechanical method, it's the enzyme. We are talking about lysing the cells, but not using sort of like mechanical force like what we had before. Okay, so in order to lyse the cells, we could use enzyme, or we could use chemical, or we could use physical force. I mean, mechanical and physical, you have to see the difference. Okay, mechanical is the actual force that does to the cell, but physical force is something else, like pressure change in concentration. I'm going to go through that in a bit. Okay, the first method of cell lysis, what we can do is the enzymatic. You probably have done some of it before. We use enzyme to lyse the cell walls or destroy the cell membrane. So basically, the enzyme that we are going to choose if we want to lyse the cell and obtain the intracellular product depends on the type of the cell wall. So different microorganism has a different cell wall structures. So you need to understand the cell wall structures of the microorganism that you want to break the cells. Okay, whether it is a lipid or peptidoglycan. Okay, that would be for the bacteria. For that type of cell wall, you have to use a certain type of the lysis enzyme. So in this case, it's muramidase or amidase. Or whether the cell wall consists of glucan, like in yeast. In that case, you probably need glucanase. Or if the cell wall are lignocellulose or pectin, which are plant cells, you might need to use some sort of cellulases, mixed enzyme or pectinase. So the knowledge on the cell wall structure is important in this case. So you can choose the right enzyme to break your cell. Okay. The second method that you can lyse the cell using non-mechanical mechanism is the use of chemical. Basically, 4 types and 3 categories of chemicals can be applied. The first one is the use of acid or alkaline. Okay, so basically, acid can hydrolyze cells, but it could cause precipitation of the macromolecules. So you need to understand as well that the products that is inside the cell, whether it interacts with the chemicals that you are going to use or not. Okay, so alkaline are more widely used than acid in cell lysis, but again, it depends on the product. You need to understand whether your product will be destroyed upon using these chemical or not. So most of the chemical are aimed to solubilize the cell's membrane. So it destroys the cell membranes and it gets something inside to release out. Okay. Another type of chemical that could be used is a detergent. A detergent would disrupt the membrane structures. So the example would be, I think, the use of tris or something when you need to extract out the DNA. So that type of thing could destroy the membrane structures as well, because the cell's membrane is phospholipid, right? So it's sort of like a micelle. So if you put some chemicals in, it alters the structures of the cell membranes, and that allows the intracellular stuff to come out from within the cell. Another type of chemicals that can be used to lyse the cells is a solvent. Okay, solvent again, it attacks membrane structures. So the chemical used for cell lysis mainly aim to either destroy the cell walls or most of the time try to lyse the cell membrane so the cell release intracellular stuff out. The last non-mechanical method are the physical method. Okay. In term of physical, we are talking the difference in concentration, so sort of like osmotic pressure. We are talking the change in temperature and those type of stuff. So physical method of cell lysis relates the use of concentrations and temperature. The first method that I want to introduce is the freezing and thawing. So in this case, you might think about when you keep the cells, store the cells in the freezer. Okay, you learn that you have to put glycerol in to a certain amount. So just make sure that the ice crystal is not too large and destroy your cells when it gets dissolved. So basically, this is a freezing thawing mechanism. The underlying things that happen is that in the cells there's a lot of water. If you don't freeze it a correct way, you get like an ice crystal to form, and when you and that ice crystal could disrupt the cell wall, the cell membrane. So that's what we want in cell lysis, but it's not what we want for storing the cells for further use. Okay, so it is the same principle. Another non-mechanical method for in the physical category is the thermolysis. Thermolysis, as the name said, is lysed by using thermal temperature. So basically using high temperature and get the membranes to collapse, okay, due to lipid fluidity. Why am I saying this? Because the cell membrane is, as you know, is a phospholipid bilayer. So with the thermolysis at high temperature, the lipid bilayer get more fluid, like moving a lot faster and tend to disintegrate. Okay, and also another physical method for the cell lysis is osmotic shock. You probably have heard it before. It is the lysis due to osmotic pressure. So osmotic pressure happens when inside the cell and the outside of the cells has so much different salt concentration. So higher concentration on one side will cause the water to go through the membrane like osmosis. So if you put the cells into like hypotonic solution, okay, so the water from outside will enter the cells and get the cells to explode. Osmotic shock lysis normally apply to the cells that doesn't have cell wall, like a very weak cells like animal cells, okay, or the protoplast, which is the yeast cell that gets the cell wall removed, so it has only the cell membrane. So basically these are the two methods, okay, two main methods to destroy the cells and get the intracellular stuff out. A mechanical method and a non-mechanical method. So you when choosing what to use, you have to weigh whether you have the instrument to do it or not, or whether your product is prone to destroy by something like heat, like some chemicals, that type of stuff. Okay, so that is about the cell lysis. Okay, there's a lot to know about. I want to give you the overall idea, some names. So if you need to do some more research, you know where to start. Okay, now comes to the main point of today's lectures is the separation of the soluble product. Okay, last time we are talking about the insoluble part, which are the cells, the solid residue. If the cell contains the products that we want, we break the cell, we got the cell debris, and then the intracellular content is released into the soluble part. So today is about the soluble part. Okay, how do we take the product that we want that is already in the fluid state, okay, in the liquid state. So in the soluble part of the product, of course we have the product and we have impurities as well. Okay, so the main aim at the beginning is to remove the impurity out. So we get higher purity product and also get it to be more concentrated as well. Okay, so before we get the product or higher purity and the final product before we go to the final form of the product. So what we are going to cover, okay, to cover is the soluble part, the product in soluble part to get the impurity out. We will looking at 4 methods, okay, 4 methods of separating the product out from the impurities, okay. These are the 4 main process that is applied, especially in biotechnology and also in other process, like in other chemical process as well. The first one that we are going to cover is the liquid extraction. Okay, the liquid extraction, it is something like this. You probably have seen it before. Okay, liquid extraction is when we employ the properties that the product has different solubilities in the different solvent. Okay, so the easiest liquid extraction is to extract with water. So if something is more soluble in water, it will go to the water phase. Another one would go to another phase. But the basic things about liquid extraction is that the thing that you want, the extractant that you want something to move to has to form like 2 different phases. The pictures that you see in this slide is from my own research. Okay, what I did was I do lactic acid fermentation, and what I'm trying to do is trying to extract lactic acid out from the fermentation broth. Okay, and normally in liquid extraction you would think that, okay, there will be 2 phases, okay, separating, but this one has like the phase number 3, so it's 3 partition extraction that could happen as well. Okay, so in liquid extraction it involves the knowledge on solubility and also the mass transfer. Okay, because you want your product to have a different solubilities in one liquid to another liquid. So that's why it can choose where it wants to go, and the solubilities of the impurity has to be different in those 2 phases as well. And it involves mass transfer because it's the move of a substance from one place to another place. Okay, it is a multi-phase process, like what you know before when we are extracting something has to involve more than 1 phase because you need different solubilities in different phase. Okay, for the liquid extraction, the 2 phases are both liquid. Okay, you can have solid extraction as well. It could be solid or liquid, or solid and gas, this type of stuff. Okay, but in biotech we involve a lot in liquid extraction. In the future you might hear something like a supercritical fluid extraction. That's going to involve a similar stuff, but in solid and supercritical fluid. Okay. So for liquid extraction, since it's multi-phase process, it involves a heavy phase and light phase. Okay, heavy phase, as you can guess, is down below, the light phase is up the top. And usually it involves the use of organic and inorganic or water phases. Okay, and three-phase extraction is possible. The driving force that makes the product goes into like the extraction process is the selective solubility in one phase over another phase. That's what I was talking before. But bear in mind that when you are doing liquid extraction, you are not doing a total separation. Okay, for example, you have two phases like this. Okay, and if your product is initially in the bottom phase, when you put the top phase and you mix them and they separate into two phases, that's going to be a little bit of your product down in the initial phase, but most of that will go to the phase that you want to extract it from. So it's not a total separation. You still have lost your product in the original phase as well. Okay. So what determines a good extraction? Okay, you see a lot of formula in this page. I'm not expecting to be able for you to be able to calculate them at this stage, but I want to introduce the term so that if you need to use extraction in your work, you could think about this term and see what determines whether your extraction that you are doing or the choice of the solvent that you choose is good enough. Okay, so I'm going to go through it a little bit. If you see the pictures here, this is a little bit clearer pictures. Okay, the heavy phase is down the bottom, the light phase is up the top, and this is a typical extraction that you will probably see the two distinct phases. Like this one is a little bit more distinct than the first pictures that you see, but you can still see like the third phase form here. So that could happen. Okay. What determines a good extraction? Three terms that you need to understand a little bit. Doesn't matter if you could calculate it or not, but at least you should understand it. The first term is a distribution coefficient. It's distribution coefficient because the product has to distribute between the light phase and the heavy phase. Okay, the distribution coefficient is normally talked into the KD term, distribution coefficient, and it's the ratio between the product concentration in the light phase over the product concentration in the heavy phase. Okay, so the concentration that we are talking about is a concentration after the extraction is done. So you start off from one phase, you add the extracted phase, and then you mix them, you leave them to separate. And that is when you determine the product concentration in the light phase or in the heavy phase. Okay, so if your product can dissolve more into the light phase, of course your KD, if you have high KD, that means that the separation is good. Okay, you don't want the KD of 1 because that means after the extraction process, the same amount of your product is in the heavy phase and in the light phase. You always want your product more into another phase, but since it's not a total separation, so you can't divide something by 0 anyway. Okay, another term that determine whether extraction is good is the extraction yield. Okay, extraction yields involve a mass balance. Okay, because you start off, for example, 10 units of a product. At the end, maybe 8 units is up the light phase, 2 units is left in the heavy phase. So the extraction yield is the mass balancing. in this case, we assume that the product is in the light phase. You see that I have concentration times volumes. So normally if you say concentration is like gram per liters or gram per milliliters, volume is liter or milliliters. When you times them together, you get a mass, okay? So basically the extraction yield is the mass that you get from the product phase. So in this case, in the light phase, divided by the whole initial things that you start off. So if you get more of extraction yield, the better your extraction is. Okay. Another term is the extraction selectivity. Okay. The selectivity means that whether it prefer this condition to another condition. You see that the selectivity is the KD term, okay, that is provided. So remember the KD is the distribution coefficient, okay, and the I and the J here is the species to be separate. So if you have your product in an impurity and you are going to use, for example, this light phase as a solvent to obtain your product. So you have to make sure that your product can dissolve more than the impurities in the light phase. So when you do distribution coefficient of your product and impurities, you get the KD for each of them, you divided them, and then you get extraction selectivity. So if I is your product and J is your impurity, of course you need your selectivity to be very high. So you make sure that your product can dissolve more into the solvent that you are choosing. So basically these three terms it can be used to determine the good extraction. Okay, and this is a common extraction apparatus in your lab. Okay, it's a separating funnel. Maybe you have seen it before. You put a thing that you mix your solvent to extract your product, and then you put them all together in this separating funnel, leave it, and then the two phases will be separated, and down below you have the valve that you can open and discard the lower phase, so you get the light phase and the heavy phase can be separated. Okay. So this is the extraction in the lab scale. Okay. This is an example of the large extraction apparatus or the machine. Maybe this is like a compact stuff, as it take a smaller footprint in the process. But for some type of extraction, you will see sort of like a big funnel separator anyway. Okay, but I just want you to have some idea that in large scale extractor, this thing can happen as well. Okay, this is called a pot-neck extractor. So basically the flow of the light liquid and heavy liquid. Light liquid is the top phase, okay? The heavy liquid is the bottom phase. They move in counter current. They move not in the same direction, a different direction. So to make sure that the mass transfer is more efficient in that way, okay? And this can be done continuously. So basically the light liquid, let's see, the light liquid comes here, okay, and the heavy liquid goes there. So the light liquid start from outside flowing in. The heavy liquid start from inside going out through these small holes, and then the contact happens, okay? And this is the large scale extractor looks like. So basically they want it to have a lot of mass transfer, but a very small footprint. So it really depends on the design, okay? Okay, that is the liquid extraction, okay? The next method or the next process that we can separate the soluble products from the impurity is the use of absorption and adsorption, okay? These two processes are different processes. However, I put them together because people could get confused between the two, okay? So I just want to talk them together at the same time so you could spot the difference and maybe understand it a little bit more, okay? So absorption and adsorption is a very similar term, but a different mechanism, okay? Adsorption is a day, absorption is a bay, okay? These pictures tells you the difference between adsorption and absorption. Adsorption is the thing that happen on the surface of the adsorbent, okay? While absorption is the media that is absorbed into the matrix of something. So it's a bulk process, while adsorption only happen on the surface, okay? Adsorption is the accumulation of the stuff on the surface, while absorption is assimilation into the bulk of something. Okay, so with the adsorption, since it is a surface, it is the accumulation of things on the surface. So at the end of the process, you will get a different concentration at the surface and the bulk. So after you do the adsorption, only things that get adsorbed is on the surface. There's nothing in this matrix here, okay? While the absorption, the matrix here absorbing the molecules. So at the end of the absorption, the concentration of the bulk matrix here and the concentration of the blue particle, maybe the product on the liquid here is, or the gas is the same. So you eventually come to an equilibrium, okay? Okay, look at the absorption first, okay? The absorption, we are not using a lot of it in biotechnology, but in the chemical process, they will probably involve a little bit more. Main application of absorption in biotech is the gas absorption or desorption. So absorption and desorption involve absorption process. Absorption is also called scrubbing, like gas scrubbing. Okay, desorption is called stripping. Okay, let's look at the scrubbing first. The absorption or scrubbing is when you have your product or something that you are interested in or impurities in gas phase. Okay, and what happened is that you absorb those particles, or not particles, sorry, those molecules into a liquid phase. That is called scrubbing. Okay, so product in gas stream get transferred into a liquid stream. The example of a scrubbing is a scrubbing of ethanol in gas stream, okay, into a liquid stream to recover ethanol loss in CO2 from fermentation. This example is very clear. Like in ethanol fermentation, in a big scale ethanol fermentation, carbon dioxide is produced, and since this is a gas, it takes away ethanol from the liquid with it as well. So if the factory just let the carbon dioxide out like that and it's a big stream, it's big gas stream, it will take away a lot of ethanol out with the carbon dioxide as well. So what they do is they have to scrub the carbon dioxide gas, so it dissolve the ethanol out into water and then going back to the process. Okay, so to recover ethanol that is lost in the gas stream. The opposite of absorption or scrubbing of the gas is the desorption step. Okay, it's another name is called stripping. This is a reverse process. So you have something in your liquid and then it gets into the gas phase. So the examples that I gave, that carbon dioxide that is produced in the liquid phase when you are fermenting, okay, and it takes some ethanol with it as well, so that's why it has to be scrubbed out again. Okay, so in stripping, the product is in liquid stream, it gets transferred into the gas stream. Okay, the application of stripping in biotech is not really the separation process to get the thing that you want, but I see a lot of people apply stripping to remove the unwanted things from their fermentation. So one example that my colleagues does for their research is the acetone butanol ethanol fermentation, and they know that all these three things, the ABE, could inhibit the microorganism growth and the ability to ferment. So they have to find some way to remove those stuff out. And since they are all volatile, okay, acetone is volatile, butanol maybe a little bit, and ethanol is volatile. So they are sparging something like nitrogen gas because they have to do it anaerobically. Sparging the nitrogen gas out and gets the volatile things out into the gas phase. So you have a less concentration in the liquid phase. That is one application that I see from the stripping. Okay, not directly into a downstream process, but you can regard it as a downstream process as well if those volatile things are your product. So you don't have to wait until the end of fermentation to do the downstream. You can just take it out while you are fermenting as well, if it inhibits your fermentation. Okay, and these are the examples of the absorption equipment. Okay, absorption. Okay, it can normally be done in three forms. Okay, the first form is the pack column. Notice that all of these three settings are column type. Okay, it takes less space and it increase the area of contact. Since if it is the mass transfer stuff, anything with mass transfer, you need a lot of surface area, you need a lot of contact time for things to happen. The first setting for the absorption equipment is the pack column. The pack column means it's a column and you pack something in. Okay, that something will give you a larger mass transfer area, and then you spray some liquid into it, and you have the gas going from the bottom. So the liquid and the gas move up, the liquid moves down. So it's a counter current flow, and the contact happen. Okay, this can be done for the scrubbing as well. Okay, another setting is the spray column. It's a similar things, but without the packing here. Okay, but for the spray column, you spray things and you have a lot of a spray distributor here. So you make sure that the gas and the liquid are in a good contact. Another setting is a bubble column. Okay, so these two, the pack column and spray column, is for scrubbing most of it, but for the bubble column is mostly for stripping. So you put the gas in and you want the gas to strip something out from your liquid. Okay, so that's why you are using bubble column. So instead of spraying the liquid, now you're trying to bubble the gas and make the bubble as small as possible. So you get a lot of surface area around the bubbles, because you're going to have many bubbles and each bubble has a lot of surface area. So in that way, the mass transfer could happen much more efficiently. Right. That was the absorption. Okay. Now looking at adsorption. Absorption is absorbed into the matrix of the thing itself, right? If it is a gas bubble is absorbed into the gas phase, and if it is a spray column, something in the gas is absorbed into the liquid phase. But for adsorption, since it is the surface interaction, okay, things doesn't get into the matrix. It get adsorbed onto the surface of what we call the adsorbent. Okay, the adsorbent is the matrix we are talking about, and whatever you want it to get adsorbed is called the adsorbate. Okay. I'm thinking of some visual example, maybe in water treatment, in the water filter. Okay, sometimes you have the resin and also the activated carbon to remove the unwanted odor or unwanted taste, the salty taste, that type of thing. You put the adsorbent in, the adsorption process happens. Okay, and the force that attract the adsorbate to the adsorbent could be physical, could be electrostatic, or could be biospecific. Okay, you can look it up a little bit more on this. Could be van der Waals force, it could be ion force, okay, or it could be like the interaction of the if you're doing something like ELISA, it is the adsorption process as well. And the pictures you see here is the example of the materials for the adsorbent. Okay, so you will only see these small beads, and these things have pores. Okay, this thing has pores inside, very small pores. So since it has the pore, that means that it has more surface for the thing to attach to. It's not just the outside of this sphere that is the surface. Inside there's a small pores as well. Okay, so these materials adsorb product or the contaminant. You can use it to separate your product, or use it to take the impurity out from your product. Okay, so either way. The desired property of the adsorbent, it is something that you have to bear in mind, that if you want to use adsorption, the adsorbent that you choose has to have a high adsorption capacity. That means that you don't use it a lot, but it can absorb so much of something that you want to recover. Okay, it has a good or high selectivity, so it's not absorbing everything. Okay, it's not absorbing and not selecting what it absorbs. So it would depends on the force as well. Okay, because some force in adsorption is not specific. Some source is specific. Okay. It should have a strong and low pressure drop design. So that means that you don't need to use a lot of driving force for it to work, because most of the adsorption process also happen in a column. If you have the strong and a low and high pressure drop. That means that it is much more difficult for things to flow through your column, okay? And it should be easily regenerated. So regeneration is the way that you reuse the solvent, okay? We are going to talk about the cycle of the adsorption a little bit later. Important concepts in adsorption, okay? Remember that these are the terms that I want you to be aware that when we talk about this term, we are talking adsorption. Just like in extraction, I want you to know those three terms. In adsorption also, okay? For adsorption, the first term that you should bear in mind is the word adsorption capacity, okay? It's very straightforward. It is how much the adsorbate, how much the thing that you want to be adsorbed get absorbed into a unit of mass or volume of the adsorbent. So if you go back to this picture, so that means you want the materials here, the adsorbent, to hold as much adsorbate as possible. So it's the adsorbate over the adsorbent, okay? The unit is depends. It could be mass per mass, mole per mass, mole per volume, mass per volume, whatever it is, but you need to understand that it is something between adsorbate and adsorbent. The higher the adsorption capacity is, the better. Adsorption isotherms, okay. Isotherms, with this word, it means that it happens at the same temperature, okay. Adsorption isotherms is how the adsorbent perform in adsorbing at certain temperature. So when the equilibrium happened, what happened, okay? This is the example of the adsorption isotherm, okay? It is actually the plot between A is the product that we want, okay? Is something that we want to adsorb. A in solution and A in adsorbent. Look back at this picture. So we want to determine at the equilibrium, means that at the end of the adsorption, how much thing are on the surface and how much thing are still in the liquid part, okay? So it is the plot between those two at one temperature, okay, at equilibrium. So in this case, you might start with, I don't know how much A you probably start off, but when you put the adsorbent in, let it go to equilibrium, you determine the amount of A in the solution and the amount of A that is attached in the adsorbent, and then you plot one plot. So this plot happens with sort of like many experiments, okay, to determine the adsorption isotherm, okay. So the process has to reach equilibrium first before you take samples, analyze, and then plot the isotherm, okay. And the common isotherms are Langmuir, Fundrich, and BET. Okay, just know the name. Okay, just know the name. There are so many more sort of isotherm. Okay, and each isotherm explain how the adsorbent get absorbed onto the surface of the adsorbent. Okay, for example, like Langmuir is a single layer adsorption. Fundrich is multiple layer and that type of stuff, okay. So different isotherm explain different thing, and it will have a different shape as well. Last but big concept in adsorption that it takes time to understand, but I'm going to go through it quickly. If you have some time to review, it will be very useful for you. It's a term called a breakthrough curve, okay? It's a breakthrough curve. Breakthrough curve is the plot between the volume of liquid that collect from the column. When I say the column, that means that the adsorption happened in the column. So the column contains the adsorbent, and then you want to adsorb something you put the liquid that contains the product or the impurities that you want to absorb, okay, and it comes out here through the column, comes out here. So you collect the volume of the liquid that exit the column, and then determine the concentration of the thing that you want in that liquid. Okay, this is a typical curve that you would get. Okay, so what happened here is that at the start of the adsorption, okay, most of the resin has not been absorbed yet. The adsorption hasn't happened yet. So everything that you put in get absorbed in the column, and then you come out having nothing. Okay, for example, if you want to remove color, okay, and if the resin is very effective, you put the color stuff in, the color liquid in, it will come out clear because all the color substance is absorbed in the column. It comes out clear, and then you put more in. Okay, you probably come out clear as well, but more of the resin is saturated already. It holds as much color substance in it already, and then you do this repeatedly. More volume you get through the column, and then at one point when all the adsorbent has absorbed to its almost maximum capacity, something is leaking out. Okay, the color start to leak out, and then the color increase, the intensity of the color increased so much, and the column or the resin cannot get rid of the color anymore. So that's the end. Okay, so this sort of curve is called a breakthrough curve. It's called breakthrough because it tells you when something breaks through the column. So like in this case, the breakthrough is here. The important things about breakthrough curve is that it tells you that in the actual operation, if you use that much resin, how much stuff that you could put in before you have to stop the operation. Okay, you could stop here if it is acceptable that the leaking is that much, or you can stop here if you accept it's okay that the leaking is that much. Okay, but definitely you don't want it until like it's full and it can't absorb anymore. So that is the idea of the breakthrough curve. Okay, it's a little bit difficult to understand the concept. Even my student has a problem with that, even though they have like the lab work and things like that. So it takes some time. If you could review it yourself, it would be very useful for you. Okay, for adsorption, a good thing about adsorption, it is the repeatable process, and you can reuse the adsorbent. Okay. For example, in your water filtration system, maybe at home if you have water filtration system, they would have some instruction like, okay, for this column, after 2 weeks or after a month, you can backflow some things and continue using the resin. So that is the adsorption stuff. So basically you start loading your column or your resin with the feed that you want to remove something out. Okay, and after you reach the breakthrough that you want, then you can start the process of regenerating it. So you pack the adsorbent in the column, and you put your feed in until what you want. Okay, then you drain the column, and then you watch whatever is not absorbed in the at the resin surface. Okay, and then what you can do is you can eluting. Okay, eluting means that looking at these pictures. Okay, eluting is that washing anything on the surface out. So the matrix here is free again. Okay, the matrix here is free again, and in order for the matrix to be used again, you have to regenerate them to the correct form before it can get reused again. So you can have this operation cycle in adsorption for many times before you have to throw away your adsorbent. Okay, you start throwing away your adsorbent when it doesn't work anymore. Okay, it has a life. Basically, it has a life, but whether how many times you can use it or when to discard is something that you have to test in the lab at least. Okay, and what will help a lot is knowing the breakthrough curve. Okay, so for the new resin, you probably could go very high for the volume for effective adsorption, but for older resin, this part of the curve will get shorter and shorter and shorter when and then it stopped working. Okay, so the breakthrough curve is a very important concept also in adsorption. All right. Okay, we have about half an hour. I think I could finish them. Okay, the next one is the membrane separation. You have heard of membrane separation. It's membrane separation is a kind of filtration, but it's through the membrane. Okay, remember we did the filtration and we used filter paper, right? We have dead end filtration and we have a cross flow filtration. Okay, and I mentioned that cross flow filtration is a normal setting for the membrane separation, and here it is. Okay, so membrane and filter, even though they probably be a sheet or a tube, you have to see them separately. Membrane are selective barrier. It based on size as well, but it is semi-permeable. Okay, it concerns the separation of small molecules, ion or particle. Okay, so in filtration, you want to take away like a larger particles, but for membranes, you are dealing with the smaller molecules, even the ions. Okay, and the membrane itself could be biological or synthetic. Okay, the classic example of biological membrane is your cell membrane. Okay, that is the biological membrane. It is semi-permeable. Synthetic membrane can made out from many chemicals, like cellulose acetate is synthetic membrane. Some sort of plastic could be made as membrane as well. Okay, what we concern on membrane filtration and what you will have heard a lot are four type of membrane filtration, membrane separation. The first one is microfiltration, ultrafiltration, nanofiltration, and also reverse osmosis. Okay, so these four are commonly known for membrane separation, and the driving force for things to happen is different, but basically is pressure, concentration, or chemical or electrical gradient. There were some other type of membrane separation as well that I'm not covering in this lecture. Is like dialysis and electrodialysis, but it consists of somewhere between NF and RO here. Now let's see the whole picture of the site separation by various type of membrane. Okay, what we had earlier about filtration is I categorize it as a clarification, so make things clear. Okay, so you clarify your broth and then you left with something that doesn't have like a large particle. Okay, microfiltration is the largest pore of all things that we are talking about here. So it covers something from, I would say 100 micrometer, 10 millimeter is very large for separation. So you start off something from 100 micrometers down to about 100 nanometer. Okay, so a droplet, emulsion, a yeast, bacteria, most of the microorganisms can be separated using microfiltration. Okay, ultrafiltration involves a smaller membrane pore than microfiltration. Okay, and most of us know already that the application for ultrafiltration is for like proteins. Okay, so ultrafiltration is sort of like a touch of nano already. Okay, and nanofiltration, of course, it concerns a nano-sized particle. Okay, and reverse osmosis is very small particle, mostly salts. Okay, and some salts, yeah, mostly salts and ions. The IEM here is ion exchange membrane. Okay, it's ion exchange membrane, and the process that involved is called electrodialysis. I'm not covering it in this lecture. Okay, you probably don't use it a lot also. Okay, if you are interested in ion exchange membrane, there are a lot of research paper on that. All right, now let's have a look at a little bit more into each of these type of membrane. So if you are separating, of course, you have to apply some sort of pressure to force the thing to go through the membrane. Even it's a dead end, or even it's a cross-flow filtration, it involves a different pressure across the membrane. Okay, so for microfiltration, it's a separation and concentration of suspended and colloidal particles, including cell. So as you see from the pictures, what is excluded from the microfiltration membrane are bacteria and suspended solid. Something else like macromolecules, enzyme or proteins, are low molecular weight substances, and water can get through microfiltration. Okay, then with ultrafiltration, apart from bacteria and suspended solid, your macromolecules like protein and enzyme is excluding as well. Okay, while the ions and other low molecular substances and water can get through the ultrafiltration membrane. And remember that each type of this membrane, it has a designated pore size. So if you want to use ultrafiltration to exclude any enzyme or proteins, you have to choose a correct sizing. Okay, for ultrafiltration, it is used in separation and concentration, also purification in this case, of the dissolved and suspended constituent. See, for microfiltration, it involves separation and concentration, but ultrafiltration, we start doing purification as well. Okay, and it based on relative molecular size or the weight. Okay, you might be asking yourself now that, hey, Ajahn, the protein and enzyme, sometimes we only refer it to Dalton, right? Dalton or kilodalton. It's actually the molecular weight. It's not the size. How do I know whether, how do I know the size of the membrane that I'm going to use? Okay, there are some sort of conversion equations, okay, that you could convert molecular weight in Dalton into the size like nanometer or micrometer. Okay, it is possible. I have a reference here for you to download. I think it's free. Okay, it's free, and you can see the formula. And there were some assumptions that is made as well, like the protein has to be globular, and that sort of stuff. You have to beware on that small points as well. Okay, the last one is reverse osmosis. Okay, you are most familiar with reverse osmosis because like all the bottled water are mostly now reverse osmosis process. Okay, so For reverse osmosis, it's a separation of water from other things, okay? Separation of water from other things, while other thing is separation of other things from water, okay? So it's separation of water from ions and low molecular weight constituent, and it involves osmotic pressure, okay? So this side will be that side, if that side is hypertonic, that side is hypotonic, and that side is hypertonic. So water get moved from where there's a lot of water through the membrane to the other side, and that is the basis um, for osmotic shock. Remember when we do this when we talk about cell lysis, okay? So it's an osmotic that happened. As you can see, if you want to separate and purify something, sometimes you have to go through a series of process. Okay, you may have to go through filtration first before you do the microfiltration, before you do the ultrafiltration, and before you do reverse osmosis. Because if you start reverse osmosis straight away, maybe the membrane get blocked by larger particles. So it designing the process is really depends on what you have in mind and the instrument that you have, and also the operation conditions as well. So that's more things to think about. Okay. And this is example of industrial scale membrane filtration system. Okay, just for you to see, because most of us will see the lab scale, okay, but the large scale is possible as well. And it could be the membrane press like this. You have a sheet of membrane frame and then press them together, or you can have like the tubular size. That could happen as well, okay, for membrane filtration system. Okay, the last method to remove the impurities, or this one can purify your product as well, is chromatography. Okay, chromatography is a basic concept we know from maybe our primary school or high school. Okay, you do paper chromatography, it's the same principle. Okay, so chromatography separation is based on a differential migration of mixture of components. So you have many things mixed together, and if you flow it through some sort of matrix, okay, some sort of phase, it will be delayed differently from by the stationary phase. Okay, the thing that move fast will go fast. The thing that moves slower will go slower. So chromatography separation involves the mixture in mobile phase. Okay, move through a stationary phase. So these two words are the keywords in chromatography. Stationary phase is something that is stay, doesn't move. Okay, the mobile phase is something that you flow through the stationary phase. And if the mobile phase contains the mixture that we want to separate, okay, those substance will move at different rate through the stationary phase, and the separation occurs. Okay, most of the separation occurs due to bonding strength between a substance, each of this color, with the stationary phase. Okay, and if you see from these pictures, you have a mixture, you flow these are in mobile phase, you flow this thing and mobile phase through the stationary phase. The blue dots move faster because the bonding strength between it and the stationary phase is the least. So it moves through the column faster. The red is a little bit slower, and the yellow is slower as well. So that's how the separation could be achieved. Okay, and the particles could be in liquid or gas phase. Okay, for the separation purpose for biotech, mostly we are involving with liquid phase. but chromatography can be both liquid and gas, and it can also apply to analytical work as well. Okay, most of you probably have heard like gas chromatography and high performance liquid chromatography. So it is a chromatography separation. We use the ability to separate by chromatography in analytical work that to determine the thing that we want as well. Okay, and if you are doing analytical work, you probably have heard the word chromatogram. Okay, it tells you like the retention time and the response, which is the curve, is for each of the component. The one that move fast will come out faster, and they have a lower retention time. The red is the second peak. The third is the yellow, the third peak. That type of thing is in chromatography separation. Okay, and in separating using chromatography, I told you that the separation occurred due to the bonding strength, okay, between the component and the stationary phase. So the bonding strength can be of many mechanisms. It can be adsorption strength, partition, ion strength, affinity, or the gel chromatography. Okay, if you are doing something about protein separation, the gel filtration is a sort of chromatography. So the gel, so it's excluding based on size as well, but it's not like the membrane that it blocked it from not going. Just that the size of the gel, the pore in the gel will slow down something. Okay, will slow down some of the component and make some component go faster, but that based on size. But it occurs due to the different moving different velocity in moving through a chromatographic column. These are the examples of laboratory scale chromatography separation. Okay, the right hand side here is HPLC column. So it's something, it's a stationary phase that is packed in the column, and then you flow your sample in the mobile phase through, and then it will get past the column and then separate into the detector. Okay, the middle part here show you the color separation. I mean, I have some research on carotenoid. We can use chromatography to separate different components of the carotenoid in our extraction as well. So like for this one, I think this one is sort of like same principle as the gel chromatography as well. So, and this is the gel chromatography column. Okay, if you are doing protein separation, you probably see this type of thing, or you can pack the gel filtration things in here, and the protein that is higher in molecular weight and size will go through first, because a smaller size will go through the pores and exit the column in a longer time. This is the example of a larger scale. So basically, if you are working in a larger scale or the factory, most of the thing you'll see the tank, you will see the column, you don't see inside. That's why you need to understand what happened inside of each of those things. Okay. Right. Okay. And that was for removing the impurities, separation, and a little bit of purification as well. Okay. So from that stage, okay, what you get here is a product of higher purity. Of course, you can make things a little bit purer by repeating those steps. Okay. And before you get the product into their final forms, you still have to do a lot of process, okay, which is considered also as a downstream process. The three process that I want to introduce for preparing the product in final form are evaporation, crystallization and drying. Okay, what I don't say here is that at the end, your product has to be in solid form or in crystal form. It could be in a liquid form as well, but more pure and more concentrate. Okay, so it doesn't have to go through all the three. Okay, you could go through one of them, two of them, or all three. It doesn't matter. Okay. Now let's see evaporation first. Okay, evaporation. Everybody knows evaporation. You put a glass of water in the room and then it evaporates. Okay, so basically it's a removal of water or solvent to make your product more concentrate. Okay, so you remove the product or solvent from the product. You are doing so by adding energy to change the physical state from liquid to gas. So in order to evaporate, the phase has to change, right? The solvent or water has to evaporate, so change from liquid into gas. In order to do so, you have to put in energy for the phase change. So you can either heat it using electricity, or if you are in a factory, you can use waste heat, because most of the factory has to have some sort of boiler, and they utilize the heat from the steam in the boiler, and they use it in different stage. So each stage that they use, the heat will lost, but still there's a lot of heat of the steam at the end of the process. You can use those waste heat steam to do the evaporation. Okay, so when the water or solvent is evaporate, okay, it will be in gas phase, right? It could be recapture, or it could be changed back from the gas phase into the liquid phase and reused again and again. Okay, and the evaporation operation involve both mass and heat transfer. Okay, so the mass transfer, think about it, is a removal of water or solvent. So we are talking mass transfer of the water or solvent. So it's a water in feed has to be equal to water in product and in vapor. Okay, so the feed that it goes in, it will be less concentrate. Okay, you remove water out, that water will go to vapor, and what is left will be in the product. Okay, so that is the concept of mass transfer. For heat transfer, the heat that you put in has to be enough to increase the temperature of your liquid. Okay, to a certain level that the evaporation could occur. So the heat that you supply is used as a sensible heat to increase the temperature, and as a latent heat, the heat for changing the phase from liquid to gas phase. So it has to have enough heat for these two things to happen. Okay. And how fast the heat transfer happens is a major controlling factor for evaporation. If you put more heat, of course, the evaporation occurs faster, but at the same time, you use more energy as well. So you have to bear that in mind. The rate of mass transfer is important when the liquid start to concentrate. Okay, that's about if you get things to very concentrate, it's more difficult to evaporate out. So that is the concept. Right? And this picture is the example of a vacuum evaporator we use in the lab. Okay, and this setting is also in larger scale as well, but it will not use in industry. Okay, so it's an effective one, and I would say if you want to use a larger scale of this setting of a vacuum evaporator, maybe it's for like high value product where you want to closely monitor things. Okay, so what happened here is you have the heating bath here. Okay, and this part, okay, this part will connect to a vacuum pump. Okay, and it creating a vacuum in the space here. So the good thing about vacuuming is that the temperature for the for phase change will drop. Okay, if that is the basic thing to do with thermodynamics. So you lower the temperature that evaporation happens if you do vacuum. This is another example of a larger scale evaporator. So basically you have the liquid in, and of course most of them use steam, okay, to heat up the tubing. So the heated tubing will sort of like boil the thing, and then the vapor coming out is a similar setting, horizontal and vertical, but the same principle. You have steam in, you have a heating element inside, and this heating element will evaporate, will make the liquid to increase in temperature, and then evaporation happen. This is another another type of evaporator. Is called a falling film evaporator. Okay, it is being used a lot in larger scale. It is the falling film because this is inside. Okay, inside is like a plating, you see, with a lot of the interchange area, the exchange area between the heat and the liquid. Okay, so what they do is they pump the product up the top, and the liquid will go down these plates. Okay, that's why it falling film. It's like when you pour water over the glass, okay, then it forms a thin film. At the same time, you put the steam, okay, through this film, the liquid film, so it evaporates more efficiently, and the product vapor, and the vapor like water goes out, and then what's left down here is a more concentrated things. Okay, and of course you can reuse the steam. That is another issue about the energy efficiency. Okay, another thing, I probably takes a little bit more time. I'm sorry about that. It's a crystallization. Crystallization is another process. Okay, that is important. Like example, the MSG is involved crystallization. Your sugar, table sugar, involve crystallization as well. I'll go a little bit quick. You can review yourself, and I'm recording this lecture as well, so you can review it afterward. Crystallization is basically the appearance of a solid crystal from a solution. Okay, it is often a final step to obtain a purified product. Okay, and followed by crystal separation and drying, then you get a final product in liquid, in solid or in crystal form. And of course, solubility is an important information that you have to have if you want to do crystallization. And crystallization occurs when a solution exceeds its solubility limits. Okay, so it has to be in supersaturated environment. Okay, I'm not explaining more about supersaturated, but I guess that you understand what supersaturated is. Okay, and it involves what we call a metastable region. Okay, I'll talk in the next slide. Okay, it's happened after the solution reaches the solubility limits and goes into the supersaturated environment. Okay, so basically no crystal is spontaneously formed in metastable region. Okay, so it's not like it's supersaturated, so the crystal form is not something, it doesn't work like that. Okay, and to form crystallization, it involves two steps. Okay, the first step, you have to have nucleation first. So you have to have something to start the for the crystal to grow. Okay, so you have to have we call a nucleus first, nucleation. Then the second step is a crystal growth. Okay, so once you have something for the substance to hold on, it start to grow bigger and bigger and bigger, increasing in size. And this is the example of the sugar crystallization. Okay, crystallization of sucrose. So what you have here, you have temperature and you have the concentration on the y-axis, temperature on the x-axis. You have the region that is called undersaturated region, and you have what called a supersaturated region. Okay, so if you go down that way, it is cooling. You go up that way is evaporation because you increase in concentration. Okay. The metastable region is the region here. Okay, it happened after the solubility limits. You see that the solubility limits, it depends on the temperature of the solution, right? Higher temperature, you have higher concentration before it gets saturated. Okay, and we have what called metastable limits. Okay, the metastable limit is after this point, you have a supersaturated region like 100%. In this region between the solubility limits and the metastable limits, there would be no crystal form. You have saturated, okay, you have saturated your solution, but no crystal form yet. So what happened is that you bring unsaturated solution in order to form the crystal. You bring the saturated solution to the metastable region, either by evaporation or cooling, doesn't matter. You take the metastable region, okay? And once it reached the metastable limits, the spontaneous nucleation will occur. So that means that a small crystal will occur by itself, okay? So with the nucleation, I'll talk about this later a little bit then. Okay, when you have a nucleation occur at this point, okay, if you maintain things in metastable region, that is when your crystal size will increase. Layer by layer by layer, it will accumulate layer by layer through mass transfer process. Okay, so everything in crystallization occurs in metastable region. Okay, and the nucleation can spontaneously occur once it reaches the metastable limits. But in industry, they don't wait until then. They add crystal seed. Okay, it's called contact nucleation. So you don't have to wait until you reach a metastable limits. You can add like a small crystal for the crystal to grow. Okay, and that is called a contact nucleation. But wait for the nucleus to occur by itself, it's called homogeneous nucleation. It takes a lot more energy and more difficult to form. Okay. Right, you see here that once the crystal grows, the concentration goes down. The point is that you have to make sure that the things are in metastable region, okay? Because metastable region, you don't have the crystal will form like from the old crystal. So you get a much more better product. Okay, so that is the main thing that happened. And remember that once you reach the metastable region, the nucleus is not formed yet until it reached the metastable limits, and that is when things would happen. Okay, this is the example of the industrial crystallizer. Okay, so it involved the refrigerant making things cool. Okay, sometimes it makes things to be vacuum as well. So everything that helps with the solubilities and evaporation, okay, and decreasing the temperature that involve the crystallization can be decided in the machine. Right, last thing, I'm sorry, please regard my sorry to your next lecture as well. So it's about drying. I'll go quick with this one. So drying is basically the final step to obtain the final product. Okay. Also involve heat and mass transfer. Drying is taking away water, but it's different from evaporation because drying is making things dry, making the moisture of the thing very small. Okay. And there were 3 drying mechanisms: direct heating, is normal. You know, direct heating, you put things in heat and then it evaporate, water evaporate. Vacuum drying, okay, it helps by decreasing the evaporation temperature. And freeze drying, okay, freeze drying is something that we also do in biotech. Okay, it involves the sublimation of water molecule under the vacuum. And one type of dryer that we see a lot is a spray dryer. I just go briefly through this. In spray drying, you can dry the liquid and make a powder. So you have something dissolved in your liquid. You feed it in the spray dryer, and then they spray it down to a small droplets, and then you flow the hot air in, okay, and the water evaporates, and then you are left with a powder product. So that is a spray dryer. Okay, so the factors are the feed flow rate, air flow, inlet temperature, and outlet temperature. That are the things that you could control in spray drying. In freeze dryer, okay, basically the product is frozen before the drying. Then the drying is done under the vacuum condition. Okay. In freeze drying, what you really want to do is you want to make the water in your final product to be ice. Okay. And then under vacuum at low temperature, low pressure, low temperature, this is a phase diagram, ice will sublime, okay, into gas, into water vapor, and then they collect water vapor here. So it's different. The other one, liquid into gas, but for freeze drying, is from ice into gas. Okay, so you have to have some knowledge about a phase diagram of water. And this is good for the product that is sensible to heat. Okay, so you can't go through a direct heat, you can't go through a spray drying, because if your product is heat labile, it will be destroyed under the high heat. So freeze drying is the way to go. Okay, this is the example of the freeze dryer. Okay, and other type of dryer as well. All right, and this is the overall concept, okay, of a downstream process. We have something probably from our fermentation. We separate the thing out, soluble and insoluble. If insoluble and it's in the cell, you do cell disruption, go through the process. You have cell debris. At the end, you have something soluble. Then you separate them using the flow method, taking out the impurities. Then you get higher purity products, and you could go through one of these 3 processes or all the 3 processes to obtain the final product.